Another cool thing about the Glagov trial was that they were able to use intravascular ultrasound imaging on coronary plaques to actually quantify the atheroma volume before and after this therapy. And by measuring that, they were able to determine that in the patients receiving both statin and PCSK9 inhibitor, that about 2/3 of patients actually saw atherosclerosis regression, and a third did still progress. However, when you look at the degree of atherosclerosis regression that these patients underwent, it was really just about 1% reduction in the percent atheroma volume. And what this is suggesting is that atherosclerosis regression in humans is probably a slow process and probably very difficult based on what the plaque is composed of. But there is hope. Earlier this year, this was published in the European Heart Journal. A 44-year-old patient presented with 70% stenosis and a plaque burden of 55% in her LAD artery. And they foregoed doing any intervention surgically and just maintained treatment with ezetimibe and statin therapy. And they saw complete regression of this plaque over the course of 3 years. So it's worth mentioning that regression can happen, but this was also a non-calcified plaque. So probably on the spectrum of atherosclerosis, one of the easier ones to get rid of. Now, when I talk about progression and regression of atherosclerosis, I like to talk about progression as just a net influx of material into the artery wall. It starts with LDL cholesterol, it includes immune cells, and then smooth muscle cell proliferation. But when we are able to correct hyperlipidemia, we then create an environment where regression can take place, and we have a net efflux of material from the cell wall. So this includes reverse cholesterol transport, as well as potentially macrophage egress or just continual cell death and efficient clearance through efferocytosis. But these are sort of the two mechanisms that I talk about. Now, experimentally, in terms of atherosclerosis regression, we have a few models at our disposal. We have the aortic transplant mouse developed by Ed Fisher's group, where you can surgically implant an atherosclerotic aortic arch into a healthy mouse and induce regression. You've got the reversa mouse, an LDL receptor knockout mouse that you can inactivate the MTTP inhibitor, and that will correct hyperlipidemia and drive plaque reductions. And finally, we have the reconstitution of an ApoE knockout mouse with ApoE, and that's sufficient to correct hyperlipidemia. So this is a model that's been used by Gwendolyn Randolph's groups and a few others, as well as we have a tweak on this where we actually use a bone marrow transplant using wild type bone marrow to restore ApoE, and I'll talk about that in just a second. So when I got to Sergio's lab 4 years ago, his group focuses on both PCSK9, but also macrophage biology and atherosclerosis. And specifically, they were looking at macrophage, a protein in macrophages called low density lipoprotein receptor related protein 1, which I'll call LRP1. And it serves as a backup receptor to the LDL receptor in the liver, but in macrophages, it actually serves a protective role during progression of atherosclerosis. And so knocking it out, you get larger plaques. We also see enhanced systemic inflammation, where over the years they detected increased circulating Ly6C high monocytes. They found increased plaque inflammation, more M1 macrophages, a higher expression of TNF alpha. Macrophages without LRP1 have increased susceptibility to apoptosis. And because LRP1 is also a pro-efferocytosis receptor, there's impaired efferocytosis and greater necrotic core production in these mice. So about the time that I arrived at Sergio's lab, we were interested in regression and how macrophage LRP1 might play a role there. And so the model that we use is we take an ApoE knockout mouse, feed it Western type diet for 12 weeks to develop intermediate lesions. And then we lethally irradiate it and subject them to a bone marrow transplant. We give them wild type bone marrow or bone marrow for mice without LRP1 and macrophages. For all intents and purposes, these mice are ApoE++, which is able to provide ApoE to the mouse and correct hyperlipidemia. We allow them to recover for 2 weeks, take down a cohort that serves as baseline, and then allow the rest of the mice to regress for 10 weeks. And when we did that, we saw that indeed bone marrow transplant with the ApoE++ bone marrow was sufficient to correct total serum cholesterol and triglycerides back down to near normal levels or near wild type levels. And when we did this, we also see that we have successfully shrunk lesion area compared to our baseline controls. And so here you can see that both wild type and LRP1 mice were significantly smaller lesions compared to baseline, and that more interestingly, LRP1 knockout recipient mice had even more plaque reductions. Characterizing these plaques, we wanted to sort of look at the inflammatory and anti-inflammatory macrophage subtypes. And we did that by looking at anti-inflammatory macrophages, co-staining CD68 with arginase 1. And you can see that, like as is you would expect with regression, significant increases in M2 macrophages in wild type and LRP1 mice compared to baseline, although no differences between the two groups. And when we look at inflammatory subtypes, the ARG2 positive cells, you see that there's actually no reduction in M1 macrophages in the wild type, but there is a significant reduction in the LRP1 knockout. So when you kind of take these two observations together, you can see that LRP1 knockout mice have a greater ratio of the anti-inflammatory to inflammatory macrophage subtypes compared to wild type. And this is due to a depletion of the M1 macrophages. We later went on to look at what could be a potential mechanism of this, and we went back to some of Ed Fisher's older work looking at the motility receptor CCR7. CCR7 was able to bind its ligand CCL19 and 21 to drive macrophage egress from plaques. And we saw during regression enhanced CCR7 expression in macrophages, but that was twofold higher in the LRP1 knockout mice. So this sort of suggests that there might be this enhanced motility when you lack LRP1 in our model here. So a quick summary of that was that the loss of LRP1 accelerates lesion progression by increasing apoptosis and inflammation and impairing efferocytosis. But the loss of LRP1 also accelerates regression by enriching for M2 macrophages and depleting the M1 population. And it increases macrophage egress from lesions. So why would the loss of a pro-efferocytosis receptor accelerate the resolution of atherosclerosis? So for the sake of time, I can't go into a lot of work, but we collaborated with Nicholas Leeper's group, and we developed a lot of cool data identifying a role for LRP1 or the necessity of LRP1 in order for anti-CD47 therapy to be effective. But we also found that we couldn't enhance efferocytosis during regression in a meaningful way. So if we can't boost regression with enhancing efferocytosis, what about the fact that LRP1 knockout macrophages have enhanced inflammation? So to get at this, well, so to touch on this, it's a little bit crazy to say, oh, enhancing inflammation must be good for regression, but inflammation isn't always bad, so it shouldn't be considered a crazy hypothesis. So many of you probably know that non-steroidal anti-inflammatory drugs, also known as NSAIDs, are actually warned, their chronic use is warned against by the FDA because they have the susceptibility to CVD events. And so here I'm just showing a couple plots where the use of different NSAIDs show higher susceptibility to MI, stroke, and cardiovascular death. And you also see in this great paper by Rahman et al., Rahman and Ed Fisher's group in 2017, they found that there is actually a necessity for this classically inflammatory Ly6C high monocyte subset and their subsequent conversion to M2 macrophages for regression to occur. And so in this paper, they blocked two receptors that the Ly6C high monocyte needs to get into the plaque, which is CCR2 and CX3CR1. And when you block that, Ly6C high monocytes couldn't infiltrate, regression did not occur, M2 macrophages weren't able to take over the plaque. So with that in mind, we went back to the LRP1 knockout macrophage, and with the collaboration with Natalie, we did a proteomic screen looking at the differences between LRP1 knockout mice and wild type mice, or macrophages. And what you see is there's actually a pretty dramatic difference in protein expression looking at LRP1 deficient macrophages compared to wild type. And over here on the right, I've highlighted some of these proteins, and a lot of these belong to pro-inflammatory or motility pathways, and that includes TNF-alpha, TLR2, and a lot of integrins. To give you an example of how these are actually changing in the LRP1 knockout mouse, this here is a volcano plot, where on the x-axis we have a log2 fold change in expression comparing the knockout to the wild type macrophage. So anything to the right of 0 is going to have a higher expression in the LRP1 knockout. And so we noticed significant increases in TNF alpha, TLR2, NOS2, and some others. And we also have significant downregulation of some other integrins, the interleukin-1 receptor antagonist protein, and also lysozyme happened to be one of the most highly downregulated proteins. To sort of validate our proteomics here, we also just ran a Western blot looking at lysozyme, and indeed, our knockout macrophages have significant downregulation of lysozyme. So, for all the non-proteomics people out there, we can kind of trust what we're seeing here based on a Western blot. So the next question is, can elevating inflammation reproduce what we find in our macrophage LRP1 knockout model of atherosclerosis regression? So to get at this, we return to our regression model, where we take an ApoE knockout mouse, give it lesions with Western diet over the course of 12 weeks, bone marrow transplant with an ApoE++ donor. And this time we decided to perform an inflammatory intervention, and we chose TNF alpha based on the fact that it came out in our proteomic screen and the long history that macrophage LRP1 has with this cytokine. So we injected 0.075 mg per kg every day in these mice, or a saline. And then it's worth mentioning that this was a shorter time point than the circulation paper that I presented earlier. So this was 6 weeks as opposed to 10. So again, when you perform this bone marrow transplant using wild type bone marrow, you're able to sufficiently correct serum cholesterol and triglycerides. And we saw no differences in serum cholesterol or triglycerides between our TNF alpha and saline treated groups. We did achieve what we think is a marker of increased systemic inflammation. We found significant increases in the spleen to body weight ratio of mice treated with TNF alpha. We saw increased circulation of granulocytes, as well as increased platelet counts in these mice compared to saline. So we think we achieved our goal of ramping up inflammation with TNF alpha. And believe it or not, the mice actually significantly regressed compared to baseline, whereas the saline did not. And there's a significant reduction in plaque size in the TNF alpha group compared to the saline. You can see this again looking at oil red O quantification, representing lipid content. And what I'm not showing is we have increases in collagen, but no difference between treatment groups, and no change in necrotic core was observed in any of the groups in this particular study. So in summary there, despite its role in protecting against atherosclerosis as a pro-efferocytosis receptor and a mitigator of inflammation, LRP1's loss in regression was able to enhance plaque resolution. Doing a proteomic screening of these macrophages, we find that the loss of LRP1 upregulates several pro-inflammatory proteins, including TNF alpha, TLR2, NOS2, and other motility proteins. And finally, by elevating systemic inflammation with TNF alpha, we surprisingly accelerated plaque regression. So this project still needs a lot of work, and we wrapped up a lot of these analyses right before COVID hit. But we hope to finish up this study once we reopen shortly. Now, I want to talk about my time here with Sergio and with Natalie. I was able to, capitalize on my reductionist biology approach, studying progression and regression of atherosclerosis with Natalie. I've been exposed to this unbiased, large omic data sets and how to analyze these experiments. And so the direction that I want to go from here as I go out on an independent scientist path is to look at epigenetic changes in atherosclerosis. And so I start with the first layer, which is DNA methylation. So many of you are probably aware that epigenetic changes can drive changes in gene expression without altering the underlying DNA sequence. And so DNA methylation occurs through certain enzymes, DNA methyltransferases that add a methyl group to the 5th carbon of a cytosine in the DNA. And if you see heavy islands of methylation, this is typically associated with suppression of gene expression. And DNA methylation has a long history with atherosclerosis. There are very specific changes that occur. Here I'm showing you a paper by Zena in 2014 where they compared human healthy vascular tissue to atherosclerotic tissue, and they see a global pattern of hypermethylation across all the chromosomes, shown here on the right in the dark blue, compared to the non-atherosclerotic tissue. And there's also been studies that demonstrate that DNA methylation can actually functionally modulate atherosclerosis progression, and vice versa. So this is a study where they took ApoE knockout mice, fed a high-fat diet for 0, 12, and 24 weeks, and they see progressing DNA methylation of microRNA 145. This is a microRNA that modulates the inflammasome. So in this study, they see that as atherosclerosis progresses, DNA methylation occurs. And they also found that an increasing inflammation can drive DNA methylation itself. So for me, breaking into this, I wanted to first detect whether or not, or first see whether I could detect differential methylation patterns in LDL receptor knockout aortas, and identify some potential gene targets that might be modulating atherosclerosis in our hands. And so this work was done in collaboration with Lucia Carbone here at OHSU. And we performed reduced representation bisulfite sequencing. This is a little bit different than whole genome sequencing. It's a little bit more targeted for CPG-rich islands. So basically, I took the aortic arch or the thoracic aorta from LDL receptor knockout mice fed Western diet for 12 weeks. We extracted DNA and performed the sequencing technique to identify how the DNA looks, or how the DNA methylation landscape looks in the diseased atherosclerotic ridden aortic arch compared to a more athero-resistant region of the aorta, that is the thoracic aorta. And one of the things we noticed was that there was actually a pretty 50-50 split between hypermethylation and hypomethylation when you compare these two tissue types. And they pretty evenly are distributed across different parts of the genome. So including the promoters, the exons, introns, and then intergenic regions of DNA. When we put our sequencing data, gosh, it's going away. When we put our sequencing data into GREAT, this is a publicly available pathway analysis software that is able to identify where DNA methylation might be affecting genes nearby. We see hypermethylation, biological processes that have to do with cell fate commitment and organ or pattern specification. And you can expect to see this based on the fact that there's probably smooth muscle cell dedifferentiation and a lot of plasticity in cells during atherosclerosis. We were also able to identify several gene families that undergo differential methylation that include several collagen genes, genes that modulate extracellular matrix, and then a lot of the classic atherosclerosis or genes involved with atherosclerosis. So we actually found hypermethylation of the ABCA1 gene. This is one of the most hypermethylated genes that we found in the aortic arch. We saw hypomethylation of LRP1 and TLR4, and some others. And so this is a cool technique that we can then use to look for some more novel targets of atherosclerosis and do a little deeper dive into understanding how DNA methylation of these genes might modulate atherosclerosis progression. But ultimately, I think I'm interested in an area of the field that probably hasn't been touched upon yet, which is what changes in DNA methylation are going to occur as extreme lipid lowering is introduced and you now have regressing lesions. Does hypermethylation of ABCA1 persist, thus making regression a little bit tougher, or do you see almost a correction of that? And so that's the path I want to go to. And so I'll wrap up there with the acknowledgements. Big thanks to my mentors, Sergio and Natalie, our lab mates, Hagai, and then Lucia Carbone and her staff, Brett Davis in the Epigenetics Consortium. And with that, I can take some questions.
Okay, well, thank you, Paul. So if anybody wants to ask questions, can you raise your hand under participants? All right.
There's also a couple in the chat if you want to look at them, Ebru.
Okay, so I'll start with the chat questions from Roberta. Roberta asks, would TNF simply increase lipoprotein lipase as explanation for plaque regression?
Increasing So that's a possibility. Increasing lipoprotein lipase would ultimately have an effect probably on the triglycerides though, which we didn't see significant changes in that. I'm not super familiar with the role of LPL within the plaque itself, so that's something I'd have to look into.
Mm-hmm. Robbie, did you want to add anything? Okay. Next question is from it's Celso Garcia Arcos. He says, or she says, I'm sorry. Do you want to say your question? The TNF treated mice are very interesting. And did you look at the body weight in these mice?
Yeah, I did. That's a really good question to ask. So the TNF alpha treated group did have so let's see, the body weight was not statistically different, but there was definitely a trend towards a lower body weight compared to the saline treated group. And they both lost weight compared to baseline. When you look at the heart to body weight ratio, there were no differences there, but there was actually a decrease in the liver to body weight ratio. So the liver is actually shrunk on these mice as well.
Oh, that's interesting. So one more question from Wendai.
Hello.
Hi.
Hey, nice talk, Paul. Thank you. I have two questions. The first question is there any, changes in the blood lipids in the macrophage LRP1 knockout mice? And the second question is that will the deficiency of the LRP1 affect the uptake of LDL by the macrophage to form the foam cell? Thank you.
Okay, those are both great questions. The first, does loss of LRP1 in macrophages affect serum lipids? The answer to that is no. There's no change in circulating cholesterol or triglycerides. And we haven't really seen lipoprotein specific changes either. Your question regarding whether there is decreased uptake of LDL.
Yes.
So LRP1's, one of its primary ligands, it has a lot, it's got like 40 plus. One of its primary ligands is ApoE. And so with that, it's actually a remnant receptor. So it takes up more VLDL or the chylomicron. And so when you knock that out, you do have decreased lipid uptake in those macrophages. But I think some good work has been shown in the field where there's actually a dysregulation of ABCA1, and you can't get it to the cell surface in these macrophages. So you actually have accumulation of cholesterol within these macrophages, even though there's slower uptake at first.
Okay, thank you.
Yep.
So I have a question, Paul. I can't raise my hand here, but so I'm interested in your bone marrow transplant model.
Yeah.
And sorry if I missed it when you were explaining it, but you irradiate the mice after they have established plaque, and then you transplant with, and they are ApoE knockouts, is that right? And then you transplant with ApoE knockout bone marrow or wild type bone marrow?
We do ApoE++. It's always going to be ApoE++ bone marrow.
Okay, so then that's what I was wondering. So then have you done it with the ApoE knockout bone marrow to control for the effects of the actual transplant and the progression? Because you sort of were comparing to baseline, but there's the irradiation and replacement of whatever the other various niches and effects. Have you compared?
Yes, so you can actually, you'll be able to see that data in the circulation paper. So that's more in the supplementary data. We did bone marrow transplant of ApoE knockout into the ApoE knockout, as well as a double knockout, LRP1 knockout, ApoE knockout into there. These mice did not regress compared to baseline. The ApoE knockout and ApoE knockout progressed just a little bit over the course of the 10 weeks. And then the double knockout actually stayed the same. It didn't. And we actually saw some pretty interesting things. The double knockout still showed a higher M2 to M1 macrophage ratio, even in the non-regression.
Okay, then just to follow up on that, maybe then, is that because the replacement of the irradiation replacement of the bone marrow, because if it's not progressing, if it's 10 weeks on a, oh wait, they're all on chow, no, you continue
They're
to
on chow diet. Yeah, they're on chow diet. And then, when you replace with ApoE knockout, the lipids drop, but they're still going to be sitting around 200 to 300 mg per deciliter. But with the addition of ApoE from the bone marrow, you can drop that back down to between 50 and 100.
Okay.
And so that's probably ultimately driving the true regression.
Okay, I think I understand. Thank you.
Sorry.
Okay, I have one last question. Are you going to look at, Paul, the hypermethylated sites in the TNF-treated mice?
That's a good question. As of right now, no. But I think it's going to depend on what I find in our DNA methylation experiments now. I'm particularly interested in the methylation patterns of LRP1 that have come up in this. But as of right now, no, we're not going to look at the methylation in the TNF-alpha group.
Because it's a little bit surprising since inflammation would drive the hypermethylation, right?
Yeah. There's some data out there from the Zong paper I showed where TNF-alpha itself can increase DNA methyltransferase expression and decrease TET expression. And so that drives hypermethylation in these. So there's definitely a correlate there for sure.
Yeah. Well, fantastic research. Best of luck.
Cool. Thank you very much.
All right. Go ahead, Katie.
Okay, so our next speaker, I don't know if you're on there. Okay, is Marie Garati, and she's an instructor at Penn in the, I believe in the CVI Institute, and will be sharing her work on, I don't actually remember the name of the title of your talk, so I'll let you take it there. There we go. Okay, thanks a lot.
Pennsylvania, and I'm going to be talking about coronary microvascular disease and how I think it's the next frontier in ischemic heart disease, which is what actually most of us are studying when we're studying atherosclerosis. So over the last 40 years, cardiologists and cardiovascular researchers have made great strides in improving mortality for ischemic heart disease. Great progress in men first and then in women. But over the last several years, there's been an uptick in mortality from ischemic heart disease. And there are lots of reasons for this. There are healthcare disparities and the rates of obesity and diabetes in this country are rising. But throughout this entire period, we've really been overlooking the role of the coronary microvasculature in contributing to the burden of ischemic heart disease. So if we want to turn this curve around, we need to think about new approaches in diagnosing, treating, and understanding ischemic heart disease. So the coronary arterial circulation is actually very complex. We usually think about it in terms of several large coronary arteries, and we overlook all of the microvessels that are a little bit smaller and not seen typically in a coronary angiogram. So it's composed of these larger vessels like the right coronary artery and these named epicardial vessels that develop coronary atherosclerosis, but it's also composed of a microvasculature. And this includes prearterioles, arterioles, all the way down to capillaries that range in size from 200 microns down. And these microvessels are actually the vessels that are physiologically active. These are the vessels that regulate flow to the cardiomyocytes, that respond to local metabolic need. These vessels are where all the action is. And whereas the larger vessels develop coronary artery disease or atherosclerosis, these smaller vessels develop coronary microvascular disease or CMVD. And we know a lot less about this process. What we do know about it is that it includes various structural changes to the microvasculature, including luminal obstruction and vascular remodeling. The capillaries can become rarefacted and you can get perivascular fibrosis. We also know that these vessels can become dysfunctional and you can get both endothelial and smooth muscle cell dysfunction in the vessels. But this process is distinct from coronary artery disease. They have some features in common. So in both cases, you get luminal obstruction, which decreases blood flow and leads to distal ischemia, and that's bad. And in both CAD and CMVD, endothelial cells and smooth muscle cells can become morphologically okay, but functionally messed up. And both processes involve multiple cell types. So endothelial cells, smooth muscle cells, immune cells, and the local fibroblasts. They also share some common risk factors like older age, hypertension, diabetes, and the usual known risk factors, but they don't share all risk factors. So male gender is actually not a risk factor for CMVD. It occurs with pretty similar incidence in both genders. There are some differences, though, that let us know that what's happening in the larger vessels is pretty distinct from what's happening in the smaller vessels, and that the smaller vessels require us to think about them specifically. And some differences are, for example, CAD has foam cell infiltration and necrotic core formation, whereas in CMVD, we don't see that. The role of immune cells is also different, although we know very little. We know that in CMVD, we see perivascular immune cell infiltration, mostly from lymphocytes. The remodeling that goes on with the vessels of these different sizes is also different, where CAD, you see outward remodeling, in CMVD, you get rarefaction, and you actually remodel by more angiogenic mechanisms because these are smaller vessels. And whereas CAD usually interferes with flow going in one direction, in CMVD, because they're smaller vessels and there are circuits, you can actually have flow in the endothelial cells coming from different sides. So clinically, this actually looks just like coronary atherosclerosis. Patients can have symptoms. There is some sort of objective evidence of ischemia. So there's either damage to the heart or stress testing that shows that there's ischemia. But when we look at the actual vessels, the actual coronary arteries, they look normal on a coronary angiogram. And so putting all these pieces together, patients are oftentimes diagnosed as having microvascular disease. So one way to think about it is thinking about ischemia in the absence of obstructive coronary atherosclerosis. Now, this is one framework to think about it, and the easiest to think about it as we're studying and getting to know more about the pathogenesis of this new disease. But it is very clear that coronary microvascular disease coexists with coronary artery disease. Some patients have one, some patients have both. And similarly, it coexists with any other cardiac pathology, a cardiomyopathy or a valve disease. It's actually really widespread, and there's growing clinical interest, which is summarized really nicely from this paper from Noel Berry-Mertz that came out in 2017 from Cedars that really highlighted the attention that clinicians are starting to give to this entity and the role of the coronary microvasculature. Similarly, in this really nice large paper from Europe, 12,000 patients that had been referred for cardiac catheterization because they had symptoms that were really convincing of angina. So when they looked at their coronary arteries, 2/3 of women and 1/3 of men actually had pretty normal coronary arteries. Yet these patients did not do well. They did not do as well as controls. They had higher incidence of major adverse cardiovascular events. So CMVD is coming out as a very important clinical entity. It contributes to mortality and morbidity. It's associated with other cardiac conditions. It's been implicated in the pathogenesis of HFpEF as well, and it plays a role in various cardiomyopathies. But we're thinking about it now and talking about it now and understanding it now because we've had technological advances in cardiac imaging over the last decade or two that are now making this field accessible to clinicians and researchers. So coronary atherosclerosis really became a huge thing when we started getting coronary angiograms and we could start to visualize lesions. Well, now we can start to have surrogate markers of what's going on at the microvascular level, which is making this more accessible to researchers and clinicians. And lastly, we really have limited evidence-based treatments and no targeted therapies for coronary microvascular disease. So it's a pretty open field for biology. So I became interested in CMVD, and the first question was, well, where do we start since we don't know a lot about it at this point? And I've always been fascinated with the idea that pathophysiology recapitulates developmental programs. So I started with the question of, well, what is the development of the coronary arteries like? And there's a transcriptional co-regulator called FOG2, or friend of GATA2, that is crucial for coronary development. If you have a knockout of FOG2, they fail to develop a coronary vasculature. And then if you conditionally delete FOG2 in adult mice, you can see what happens here. The microvessels recede and you get rarefaction and inadequate perfusion of the heart compared to a normal mouse. And the mechanism of this is that FOG2 is actually regulating a proangiogenic program in concert with GATA4, not just in development, but it looks like it's also very important in adult mice to maintain the microvasculature. So we wondered if FOG2 might play a role in human coronary microvascular disease and not just be important in mice. So in order to see if that was the case, we actually used a genomics approach. So just like for genome-wide association studies, we can compare a single phenotype and see if there are any genetic associations across the genome that might be associated with that phenotype. We can do kind of the converse of that and do a PheWAS, or phenome-wide association study, and look to see if there's a genetic variant or a group of variants that are associated with various phenotypes, or in this case, clinical diagnoses. So we used the Penn Medicine Biobank, which consists of about 11,000 participants on whom we had genotype and whole exome sequencing data and electronic health record data. And we actually just asked the simple question of, if we look at FOG2 variants, do we see any signal? And so we looked at 34 non-synonymous SNPs that were in the FOG2 coding region and performed 34 PheWAS analyses. And of these, one showed a really interesting finding. So one variant, RS283, was associated with the human diagnosis of atherosclerosis. So we looked a little bit deeper to see what this variant did, and this variant coded for an A to G substitution at base pair 1968, which results in a serine to glycine substitution. And the FOG2 protein is pretty big, and this change was actually happening kind of in the middle in this loop area. And what's interesting about this variant is that it actually has been reported to be associated with complex or conotruncal congenital heart disease in humans. We thought that was pretty neat. What was even neater is in our patient population, we had 44 patients that were homozygous for this variant. We had 500 heterozygous patients and about 10,000 controls. This represents a minor allele frequency of 0.03 in our biobank, which is similar to these large databases. But what you'll notice is that if you look at databases that are exclusively of participants of African ancestry, the minor allele frequency is much higher. And actually, of our 44 patients, 41 of them were African American. So it's a variant that's actually pretty African American specific. And as an aside, when we looked for congenital heart defects, we actually found 2 of our 44 adults that had complex congenital heart disease that had been repaired. So this did reach statistical significance and suggests that the variant is indeed associated with complex congenital heart disease, but it's also existing in many adults that are walking around with no congenital heart disease. In order to phenotype these patients and understand why we're getting the signal for atherosclerosis with this FOG2 variant, we used the electronic health record data to determine whatever parameters we could. So we looked at coronary angiograms, we looked at echocardiograms, and to just skip to the punchline, we saw no difference in left ventricular structure or function and no difference in hemodynamics for these patients. But what we did see when we looked at the different genotypes is that patients that had the GG genotype actually came into the emergency room with chest pain more frequently. So they were presenting with chest pain syndromes and getting admitted more frequently than the heterozygous patients and more frequently than the homozygous, than the control patients. We then looked at our 44 homozygous GG patients and developed some matched cohorts so that we could do deeper phenotyping. So we matched based on age, gender, and race. And you can see that the homozygous patients were about 58 years old, 61% of them were female, and they were mostly African American. So when we matched based on these, we actually had a very similar risk profile, risk factor profile for other common risk factors. We looked for the burden of coronary artery disease in these cohorts based on coronary angiograms and actually saw that GG participants had less coronary artery disease. So they were coming into the emergency room more frequently with chest pain, but they had less coronary artery disease. We then looked at clinical criteria for CMVD, kind of what I showed before, which is symptoms consistent with ischemia, but a coronary angiogram showing no obstructive CAD. And when we looked at patients that met these criteria, we actually saw that the GG participants met these criteria much more frequently. And the last way we assessed the coronary microvasculature was by looking at their coronary angiograms and determining something called the TIMI frame count. So you can see that in the right panel, the contrast dye seems to be moving through the coronary arteries pretty slowly. It's a little bit more sluggish, whereas in the left panel, it courses right through. So if you do not have a proximal stenosis, the rate at which the dye is flowing is actually reflective of resistance at the microvascular level. So Mike Gibson in the 90s actually developed the TIMI frame count, which is a quantitative or semi-quantitative measure of how long it takes the dye to flow from a proximal landmark to a distal landmark. And when we looked at our cohorts, we actually saw that the GG participants had altered myocardial blood flow and altered flow rates using a TIMI frame count. So this was pretty compelling evidence that that FOG2 variant might play a role in human adult vascular coronary disease. We wanted to gain a little bit of insight into what this variant could be doing. So we looked at some peptide modeling, and this serine is actually part of this loop right here. And it forms a rigid loop. And if you swap it out for a glycine, you introduce a lot of mobility and a hinge point here. So you can get changes in secondary and tertiary structure here because of this one base pair mutation. This variant is not in one of the key zinc fingers or regulatory domains. It's kind of in the middle. And so we thought maybe that's why it's changing the protein, but not completely ruining its function. So we overexpressed FOG2 and the FOG2 mutant in a cardiomyocyte, a human cardiomyocyte cell line to try to understand the effects. And at first glance, we actually saw no difference. We saw no difference when we overexpressed FOG2 or the mutant in large numbers within a CMV6-driven overexpression system. And while this was frustrating, it actually was pretty interesting because it's telling us that FOG2 as a transcriptional co-regulator isn't doing much on its own. It needs a binding partner. So based on the fact that FOG2 is expressed in the same time frame in development as HIF, and it actually shares a very important target of HIF in VEGF2, we hypothesized that FOG2 could serve as a cofactor for HIF. So as many of you know, HIF is a very well-studied protein. In the setting of hypoxia, the HIF protein is stabilized and it leads to transcription of angiogenic genes, and it also inhibits mitochondrial function. In AC16 cells, this mechanism is intact. When we expose these cells to hypoxia, we see down here that the HIF protein builds up over time, and that leads to transcription of known HIF targets. Angiogenic targets like VEGF are transcribed more, as are metabolic targets of HIF, like PDK1 and BNP3. So we asked the question if FOG2 affects how HIF does its job in these cells. So to test this, we used CRISPR-Cas9 to generate FOG2 knockdown cell lines, not knockout, but knockdown. And you can see that we were able to decrease the amount of FOG2 in these lines by about 50% or 75%, and that the FOG2 protein was also decreased. And what we found when we exposed these cells to hypoxic conditions is that the control cells behaved as predicted, where over time you get increased transcription of known HIF targets like VEGF-A, adrenomedullin, and ANGPT. The knockdown cell lines had a blunted response. So VEGF transcription did increase, but it increased much less so than the controls. The same was true for adrenomedullin and ANGPT1, and these are all angiogenic targets. But when we looked at metabolic targets like PDK1, BNP3, and COX4-1, we saw that there was no difference between the controls and the FOG2 knockdown cell lines. So in other words, it looks like FOG2 affected how HIF regulated transcription of angiogenic genes without having much effect on the metabolic targets. So knowing this, we then went back to our FOG2 variant and asked the same question, what happens when we overexpress FOG2 or the FOG2 variant, but now did this under hypoxic conditions? And then we started seeing a lot of differences. We saw that VEGF and ANGPT1 and adrenomedullin were differentially expressed between FOG2 and the variant. And then we took conditioned media from these hypoxic cell cultures expressing FOG2 or the FOG2 variant. We saw that the protein in the conditioned media was different. So there were different levels of ANGPT1 and other proangiogenic factors. And then when we took that conditioned media in that soup and put it on Huvex and Matrigel, we found that the FOG2 variant was actually able to promote angiogenesis more than the FOG2 samples. So to summarize, CMVD contributes to the huge burden of ischemic heart disease and is an understudied area of coronary vascular biology that has huge clinical implications. We studied FOG2 because of its role in development and found that FOG2 is not only crucial for coronary development, but FOG2 variant A1968G is associated with human adult disease, and therefore FOG2 may play a role and may be important for human adult disease. We found that FOG2 modulates a HIF1 induced expression of angiogenic genes like VEGF-A and adrenomedullin, but not metabolic targets like PDK1 and BNP3. And we also found that more specifically, the variant of FOG2 that we were interested in altered HIF-induced angiogenic program. And here is an overview of a model that we think explains what might be going on, that in the setting of FOG2, when you turn on the HIF pathway, FOG2 affects which targets are expressed. So whether you transcribe more of these angiogenic targets or whether you have more of a metabolic effect. So you can manipulate that based on how much FOG2 is available. And then these factors leave the cardiomyocyte and signal to the microvasculature to either encourage it to grow and support it and mature the vessels, or if you don't get the right soup of these growth factors, then you get a dysfunctional microvasculature. So I want to thank everyone that was involved in this work, and I'm happy to answer any questions.
Sorry about that. That was really excellent, Marie. Thank you so much for that really beautiful overview. Maybe I'll take the first question because I'm moderating. I'm wondering, maybe I missed it. So FOG2 is obviously required for the microvasculature program, but is it expressed by any other cells during any other, whether it be pathological or normal processes?
Yeah, I think what's interesting is that it's expressed in the heart, which made it really interesting to me. It's also expressed in cerebellum and in gonads. So the main tissues of expression are actually those. It is expressed in endothelial and smooth muscle cells, but the reason we chose to focus on cardiomyocytes is because in the JCI paper from Bill Pugh's group, they actually conditionally deleted FOG2 from endothelial cells, smooth muscle cells, and cardiomyocytes, and only saw an effect when they took it out of the cardiomyocytes. So it is expressed in these other cell lines, and it probably plays a role, but it may not play as dramatic a role as what we see in cardiomyocytes.
Okay, excellent. Okay, so there are a couple of questions from the chat, and I also encourage everybody, you're welcome to use the chat if you feel the best doing that. Otherwise, just raise your hand, and I'll come to you. Okay, so the first question is from Ernesto Pina. Did you try studying FOG2 mutants in other human cell lines? And you sort of implied that in your last question,
Yeah,
such as
I think we focused on the cardiomyocyte for that reason.
Okay, great. So from Mete, are there any other variants in high LD with your variant of interest in people with African ancestry?
So we've looked at some of the variants that are nearby, and there are some, so it's a little bit hard to tell for the patient data if it's specifically this variant that's causing it, or if there is a different variant that's nearby that's responsible for it. But I think some of the in vitro functional data suggests that this one specific base pair is doing something that might be relevant. But clearly that speaks to the point that we need to look at this in a mouse model, and that's one of the things we're looking at to see if this variant specifically affects physiology.
Okay. So do you envision FOG2 overexpressing iPSCs differentiated as a novel therapy for MI?
I think that's one potential way of looking at it. One of the things I found very exciting about the negative data that I showed first is that we could overexpress FOG2 and actually get very minimal gene expression change. So though you would never want to mess with HIF or GATA4 in a cardiomyocyte in a human, you could potentially manipulate FOG2 and then only have gene expression changes under hypoxic or ischemic conditions. So I haven't thought necessarily about using iPSCs with it, but actually FOG2 directly might have therapeutic potential.
Okay, the next question is from Laura about the expression of GATAs 4, 5, and 6 when you modulate FOG2 expression.
Yeah, that's a great question. And we, so GATA was expressed at very, very low levels in these in vitro cardiomyocytes, and understanding how FOG2 interplays with both HIF and GATA is kind of the next step. I think the novelty in this is showing that it can affect how HIF does. It's a known partner for GATA4, but it's not known that it's a partner for HIF. So it'll be interesting to understand how it interacts with both transcription factors.
Okay, excellent. And from Ernesto again, did you look at FOG2 modulated HIF1 expression in the SVF, more specifically innate immune cells, macrophages, et cetera?
So no, I haven't really looked at, I am not an immunologist by any stretch of it. And there's clearly a signal that there are some changes in some signaling molecules that are very important for the immune cells, but no, I haven't looked at that specifically.
Okay. And does CMVD precede coronary atherosclerosis from Carlota Oliega?
I think they're two completely separate processes. I think if you have some systemic insults like hypertension or diabetes, depending on your genetic and epigenetic environment, you may get atherosclerosis or CMVD or both. But I think they are two distinct processes, just like aortic valve sclerosis involves endothelial cells and the valve interstitial cells, but it's different. Some patients have valve sclerosis, some have CAD, some have both. I think about it the same way. They're two separate processes that have a decent amount of overlap.
Sorry, I'm being interrupted here. What did I lose here? From Paul, do you have scope the scope to call patients back and quantify
Yeah.
by proper CMR?
Yeah, that's a great question. Actually, I didn't show the data, but I have my by PET. I read nuclear studies, and we have a large program here where we do flow perfusion in these patients. And there is actually a difference in cardiac PET determined perfusion for the different genotypes. So I didn't show that data, but we do have for using PET.
Okay, the next question from Shruti of whether or not you grew your cells under hypoxic environment to see if the supernatant still promotes tube formation.
Yes, it was only in hypoxic environments that it promoted tube formation.
And then whether or not the CMV, any effects by metabolic syndrome or any other organ such as the brain.
Yes, I think the, so does it affect the heart globally? Yes, it does. It can cause ischemia directly, cause myocardial infarction. There's also growing data that it can lead to heart failure with preserved ejection fraction, which is a problem in diastole or the heart's ability to relax. So it definitely affects the heart globally. There is an effect of metabolic syndrome on CMVD. It's clearly a huge risk factor. Diabetes and obesity damage the microvessels. And there is some overlap in pathogenesis with what's going on in the brain. We know very little about kind of how they're similar or not, but I will say that FOG2 is also expressed in the brain. So it's the brain, the heart, and the gonad. So that's another potential area of interest.
Excellent. Okay, well, thank you, everybody. If there are any other questions, and raise your hand, last chance, if so. If not, okay, then we will move on to our last speaker, Lale.
Okay, so the last speaker is Lale Özkan, and Lale is a very talented PI at Columbia University. She's an expert in glucose and cholesterol metabolism in the context of diabetes, obesity, maybe also a little bit of atherosclerosis. And she's going to be the next presenter and the last presenter this evening. So Lale, the floor is yours, or the screen is yours. I don't know what to say in these steps.
Hey,
Lale,
great.
do we see you? Yeah.
Thank you, Esther. Wonderful. You can see it, right?
Yes.
Okay. Wonderful. Great. So thanks for everyone to stay this late. We'll switch the gears. Up to now, we talked a lot about vascular wall and heart. Right now, we're going to talk about processes that originate in the liver and contribute to cardiovascular disease. So as you know, upon fasting, glucagon is secreted from pancreatic alpha cells, and it mostly acts on the liver to acutely increase blood glucose. And this is a mechanism to prevent hypoglycemia. And it's been shown that glucagon also regulates amino acid and lipid metabolism. But this response has been shown to be aberrantly activated in type 2 diabetes. Type 2 diabetic patients have been shown to have uninterrupted glucagon secretion, meaning upon hyperglycemia, they can still secrete glucagon. And it's been shown that they also have high circulating basal glucagon levels. Because of its hyperglycemic effects and high circulating glucagon levels, there has been a lot of interest in inhibiting glucagon action as a treatment for diabetes. The companies back in early 2000s started synthesizing glucagon receptor antagonists to treat type 2 diabetes. And most of them reached phase 2 clinical trials. For example, this one compound, MK0893, reached phase 2 clinical trials. And in type 2 diabetic patients, it was able to nicely lower fasting and postprandial plasma glucose and hemoglobin A1C. However, this came with a side effect that upon these reductions, upon inhibiting glucagon receptor, plasma LDL cholesterol levels were increased. And this was a significant block for their development as type 2 diabetes agents. In fact, if we look at the earlier studies in mice, we can see the same phenomenon. For example, here you can see that in glucagon receptor deficient mice, compared to wild type mice, you can see that they also have high circulating LDL cholesterol levels, suggesting that there's definitely a link between glucagon action and cholesterol metabolism. But the underlying mechanisms are largely unknown. So we became interested in this link between glucagon and cholesterol metabolism. And we wanted to attack this by a genetic method. We wanted to acutely block glucagon action, just like what we do in humans, using adeno-associated virus 8, where it contains an shRNA targeting glucagon receptor that is driven by H1 promoter, and it's been shown that this adenovirus mostly targets hepatocytes. So we took both obese and lean mice, which I'm going to talk later, and treated them with control or SH glucagon receptor to block glucagon action. And we were able to nicely lower glucagon receptor mRNA levels in the liver. And as expected, this led to lowering of blood glucose levels when we inhibited glucagon action. And again, similar to mouse and human studies, this led to an increase in plasma total cholesterol. And when we fractionated this cholesterol, these are wild-type mice. As I said, it's just that we block their glucagon receptor. And as you can see, the LDL cholesterol, which is normally very low, was significantly increased when we block glucagon action, suggesting that hepatic glucagon receptor silencing in this model recapitulates what has been shown in humans and in previous mouse models. So we decided to understand the mechanism. Since LDL cholesterol is mostly regulated by the expression of LDL receptor protein in the liver, we next checked LDL receptor protein in the liver. And compared to control, we observed a significant lowering of LDL receptor at the protein level. So in order to understand the mechanism, we then checked its mRNA. And surprisingly, the LDLR mRNA was not lower. And in fact, in most of the experiments, we observed significant increase in LDLR mRNA, perhaps due to compensation. So suggesting that blocking glucagon action regulates LDL receptor at the protein level. So in order to understand what pathways might be regulating this, we turned our attention to well-known target of LDLR regulator, which is PCSK9. As you know, in 2003, first mutations in PCSK9 were described to cause hypercholesterolemia. And then since then, its mechanism of action has been uncovered by many labs, such that the secreted PCSK9 protein binds to LDL receptor at the plasma membrane and increases its lysosomal degradation, thereby preventing its normal recycling, so that an increase in PCSK9 levels lowers LDLR protein, which leads to an increase in LDL cholesterol. Despite its well-known action of how it regulates LDL receptor, and as a therapy recently more than before, the mechanism of how PCSK9 is regulated is largely unknown. So with this background, we checked whether PCSK9 levels change in mice where we inhibit glucagon action in the liver. And we observed a twofold increase in circulating PCSK9 levels in these mice. Since liver is the major organ to secrete PCSK9, then we next check PCSK9 protein levels in the liver of these mice. And similar to plasma cholesterol, the PCSK9, we observed a significant increase in PCSK9 protein in the liver of these mice. So up to now, we had data suggesting that when we block glucagon action, we observe an increase in LDL cholesterol and a parallel increase in PCSK9. But we didn't know whether the increase in PCSK9 was the reason for why we see increased LDL cholesterol in these mice. So in order to answer this question, we silenced PCSK9 together with glucagon receptor. And as designed, you can see the plasma PCSK9 levels in this double SH transfected mice was significantly lower than SH glucagon receptor alone and closer to wild type levels. And under these conditions, we asked what happens to LDL cholesterol. Again, in this experiment, we were able to reproduce the increase in LDL cholesterol by glucagon receptor deficiency. And when we inhibited PCSK9 in this background of glucagon receptor deficiency, we were able to lower LDL cholesterol back to wild type levels, suggesting that glucagon receptor inhibition increases PCSK9. And this is the reason why we do see a change and increase in LDL cholesterol. We also did these same experiments in PCSK9 knockout or LDLR knockout animals where we inhibited glucagon receptor in the liver, and we got similar results. Namely, there was no change in plasma LDL cholesterol, suggesting that glucagon acts through PCSK9 and LDLR to regulate LDL cholesterol. Next, we asked, up to now, we were using a little bit an artificial model where we were inhibiting glucagon action. Next, we asked whether in treating these mice with glucagon or whether glucagon has an effect on PCSK9 or cholesterol levels. So we treated, in order to understand this, we treated wild-type mice with glucagon for 3 days. This is important because after 3 days, we know that glucagon also lowers body weight, and which could have an impact on circulating cholesterol levels. So for 3 days, we treated them 3 times a day with glucagon without a change in body weight, and we were able to report the increase in circulating glucagon in these mice. And as expected, PCSK9 levels were significantly lowered. And this reflected as a lowering of LDL cholesterol, as you can see, without a change in other cholesterol subtypes like VLDL or HDL. Okay. We also did the same experiments in PCSK9 deficient mice. We treated them again with 3 times a day with glucagon for 3 days. And again, without a change in body weight, we were able to increase circulating glucagon levels. And this led to no change in circulating cholesterol levels. And same thing we observed in LDLR deficient mice, where we didn't observe any change in cholesterol. I'm not going to show all the data, but with our collaboration with Regeneron, with Haruka Okamoto and Jesper Gromada, we obtained more in vivo proof using different genetic models showing that hepatic glucagon receptor signaling regulates PCSK9. The data so far I have shown you was using diabetes obese mice, but we also observed that when we silence hepatic glucagon receptor in both lean and genetically obese mice, in OBOB or DBDB mice, we observed an increase in PCSK9 and plasma LDL cholesterol. Also, when we treat both lean and obese mice with anti-glucagon receptor antibodies, there was an increase in PCSK9 and LDL cholesterol. And lastly, we observed that glucagon deficient mice also had increased PCSK9 and plasma LDL cholesterol levels, showing that using different genetic models and different mouse models, we were able to show that hepatic glucagon receptor signaling regulates PCSK9. So next, in order to gain more insight into the mechanism and to understand whether this is cell intrinsic to hepatocytes, we moved to an in vitro model where we silence glucagon receptor in isolated hepatocytes. And similar to our results in vivo, we observed an increase in secreted PCSK9 in these isolated hepatocytes. Conversely, when we treated freshly isolated hepatocytes with glucagon, we observed, similar to our in vivo results, that glucagon treatment lowers PCSK9, suggesting that these are originating in the liver and affecting the whole metabolism. So next, we wanted to understand more mechanistic insight of how this is regulated. And then, as you know, glucagon, upon binding to its receptor, on one hand, activates phospholipase C and increases intracellular calcium. And on the other hand, it activates adenylate cyclase, where it increases intracellular cyclic AMP. So we asked whether this canonical target of glucagon receptor signaling, an increase in cyclic AMP, is responsible for the changes that we observe in PCSK9. So in order to understand whether cyclic AMP is responsible, we treated primary hepatocytes with forskolin and adenylate cyclase activator, or dexamethasone, which inhibits degradation of cyclic AMP. And we observed that this treatment with forskolin and dexamethasone resulted in lowering of PCSK9, similar to what we observed with glucagon treatment. And this also resulted in lowering of secreted PCSK9, which also showed as an increase in LDLR protein levels without a change in its mRNA, suggesting that cyclic AMP is the downstream signaling factor in glucagon receptor signaling. Next, we asked whether this canonical cyclic AMP target, PKA, is responsible for these effects. In order to understand whether PKA has a role, we inhibited PKA using specific PKA inhibitors. One of them was HOD9. And the idea was whether blocking PKA action would rescue the lowering of PCSK9 by glucagon treatment. To our surprise, we didn't see a change in PCSK9 levels when we inhibited PKA, suggesting that PKA is not involved in this. So after excluding PKA downstream of cyclic AMP, we turned our attention to another cyclic AMP target, which is less well studied compared to PKA, and it's much more studied in other cell types, but not in liver, and which is the exchange protein directly activated by cyclic AMP, EPAC. And it's a guanine nucleotide exchange factor for small g proteins like RAP1 and RAP2. So we asked whether EPAC2 is our target downstream of glucagon receptor signaling. We silence EPAC2, and similar to silencing glucagon receptor, we observed an increase in PCSK9 in primary hepatocytes. And conversely, when we treat hepatocytes with a cell-permeable specific EPAC2 activator, HCP-TOM, we observe a small but significant lowering of PCSK9, similar to glucagon treatment, suggesting that cyclic AMP regulates PCSK9 through EPAC2. After that, we wanted to understand what is downstream of EPAC2. And as I said, RAP1 is a small G protein that gets regulated by EPAC2. And it's not well studied in the liver, but it's being shown that it's required for actin cytoskeletal organization and signal transduction in other cell types. So we asked whether RAP1 is our molecular target in this pathway. When we silence RAP1, similar to silencing of glucagon receptor or EPAC2, we observed an increase in intracellular PCSK9 levels in primary hepatocytes. So very briefly, how RAP1 gets regulated, as I said, it's a small G protein, and it's activated when it's GTP bound form, and that is mediated by EPAC2. And it's inactivated when it's GDP bound form, and that is mediated by GTPase activating protein specific for RAP1, which is RAP1 GAP. So another way of inhibiting RAP1 is to overexpress this inhibitory protein, RAP1 GAP. We had an adenovirus through our collaborator, and when we inject this adenovirus expressing RAP1 GAP, we observed an increase in plasma PCSK9, similar to glucagon receptor inhibition. And this led to lowering of LDLR protein levels without a change in its mRNA. And that resulted in an increase in both plasma total cholesterol, and when we fractionated, we observed that this was mostly LDL cholesterol, suggesting that RAP1 in vivo recapitulates what we see when we inhibit glucagon receptor, namely an increase in plasma LDL cholesterol. So we asked the question whether if we can forcefully overexpress activated RAP1 in the liver, whether we can lower PCSK9. And in order to do that, we overexpress this mutant form of RAP1, which is the Q63E mutant that is lacking the binding site of RAP-GAP protein. Therefore, it's constitutively GTP bound, and therefore it's constitutively active. And when we overexpress this by injecting adenovirus expressing CA-RAP1, we observed lowering of PCSK9 that led to an increase in LDLR protein levels without a change in its mRNA. And that resulted in lowering of plasma total cholesterol. So what we have shown as a summary is that glucagon, upon binding to its receptor, increases cyclic AMP, and cyclic AMP through this EPAC2-RAP1 pathway somehow regulates PCSK9. And the molecular mechanism we studied was that PCSK9 gets regulated at the protein level. During the course of our experiments, we never observed a change in PCSK9 when we either activated or inhibited this pathway, suggesting that somehow glucagon receptor signaling regulates PCSK9 at the protein level. And we believe lysosomal degradation is important in these processes, suggesting that glucagon activation of glucagon receptor signaling somehow enhances lysosomal degradation of PCSK9, because when we block lysosomal degradation, we were able to rescue the lowering of PCSK9. So these are my summaries suggesting that using different mouse models, genetic mouse models, and differentially different fat mice, obese mice, or diet-induced or child-fat mice, we were able to show that hepatic glucagon receptor increases PCSK9 by an EPAC2 cyclic AMP mediated pathway. And we believe this is through its effects on PCSK9 degradation. So right now, what we're doing is that we are trying to understand what other regulators of this pathway might be important for PCSK9 regulation, such as RAP1, how it gets regulated by other signals other than glucagon. So there are some preliminary data, but it's too preliminary to talk about. But what we have shown is that, as I said in the beginning, the regulation of PCSK9 is largely unknown. And we believe in terms of hormonal regulation of PCSK9, we believe glucagon receptor also participates in its regulation, thereby contributes to cholesterol metabolism. So with that, I will stop here, and I'd like to thank all my lab members and my collaborators, Erika, Jesper, and Haruka, and I am ready to take any questions.
Yeah, thank you, Lale, for this very clear presentation. I see some questions in the chat. The first one is from Katie. Katie asks, wondering if you have checked whether the elevated PCSK9 in the glucagon receptor deficient mice is physically bound to LDL particles.
Yeah, Katie, that's a great question, and I get this question almost every time because the ELISA that we're using does not differentiate whether it's LDL bound or it's free or unbound. I would like to know that. I know Regeneron, I guess, has an ELISA which can differentiate, but the short answer is I don't know.
Okay, next question from Natalie. She says, great data. It's hard to think about glucagon metabolism without insulin in the picture, specifically with, specifically with PKA. My chat is moving up and down. With PKA signaling involved, do you know how insulin-glucagon interaction contributes to the overall LDL receptor regulation?
Yeah, that's a great question, Natalie. What I can tell you is there's definitely, PCSK9 definitely gets regulated by insulin, and we believe the regulation of PCSK9 by insulin is at the mRNA level. We know that. It's been well studied by others. Coming back to our data, we know that at least in this model where we block glucagon action, insulin levels do not change. So suggesting that if insulin is out of the picture, you know, glucagon definitely has a role. But when you think about the overall metabolism, I think it depends on whether the fed or fasted status of the mice, you know, whether insulin wins or glucagon wins. But things change in perhaps in type 2 diabetes where there's where glucagon cannot be suppressed, you know, so definitely it gets differentially regulated by insulin and glucagon, yes.
Okay, another question from Paul Carter. Is there any work for combined glucagon and PCSK9 inhibition as a likely very expensive treatment for diabetes, hypercholesterolemia, and cardiovascular risk?
Yeah, that's a great question. Right. So yeah, that would be ideal, right? Like when we can combine glucagon receptor inhibition, where we can safely lower blood glucose, and we can get rid of the side effect of glucagon receptor antagonism on plasma cholesterol levels by also silencing PCSK9. So that's possible, perhaps in the future, but right now, I don't know if any company is working on it. Yeah, that's a great one.
Okay, are there any more questions? Please raise your hand or start typing. No. Lale, I have one last question. I'm not really familiar with your field, but one of the other candidates for PCSK9, another molecule with a similar functioning, is the molecule IDOL. have you looked at idle levels, and does that play a role? Like,
Yeah, that's a great question. When we initiated this work, we also checked idle levels, but as far as I remember, we did not observe a change in idle, and therefore we focused mostly on PCSK. But that's a great question. Yeah.
okay, well, thank you very much. Then I think this concludes our session. And Abel, do you want to close?
Sure. Well, thank you, everyone. So we'll see you next week, Friday. Thank you, Lale. Thank you, Paul. And thank you, Marie.
Thank you, everybody. Great to see everyone. See you
So the
later.
organizers will come back after everybody leaves. All right. Bye, everyone.
Bye.